Good afternoon, everyone. On behalf of whole Manav team, I welcome all of you for today's webinar in the Data Science webinar series organized by Manav Human Atlas Initiative. Myself, Dr. Saleh, moderator of today's talk. And as you all know, that the total allotted time for the webinar is 1 hour, out of which 40 to 45 minute talk will be presented by speaker. And we'll have a 15-minute question answer session at the end of the talk. I request all participants to use Q&A tab at the bottom of your screen to put your queries. You can put your queries in between the talk, but it will be answered by the speaker at the end of the session, means after the talk. I request again to use Q&A tab at the bottom of your screen to put your queries. It's my pleasure to introduce Dr. Vibhor Kumar, speaker of today's webinar. Dr. Vibhor is currently working as Associate Professor in Computational Biology at Indraprasth Institute of Information Technology, that is IIIT-Delhi. He has completed his PhD in Computational and Systems Biology from Helsinki University of Technology, Finland. He has completed his master's as MTech in Computer Technology from IIT-Delhi. And he has worked with computational problems for analyzing large image data set from cryo-EM and statistical analysis of genomic data. During his PhD, he has solved first and the high resolution structure of LDL, the bad cholesterol, in the native state of human body temperature. After completing his PhD, Dr. Vibhor started applying computational approach to genomic data and worked for 1 year at Personal Genomic Company in Malaysia. He has worked at Genome Institute of Singapore, GIS, for more than 8 years and has published some fundamental research findings. At GIS, he was primarily working on epigenome and gene regulation and applied his finding to other branches of science, like cancer and stem cell studies. At IIIT Delhi, his group has taken the lead in the field of single cell epigenomics. It is really great to have Dr. Vibhor with us for today's webinar, and he will be talking about the analytical challenges and promises in single cell epigenomics. Now, without taking too much of time, over to Dr. Vibhor.
So I'll share my screen. Okay, and
yes.
Yes.
Yeah,
so I hope everybody can see. So I am Dr. Vibhor Kumar. Okay, this is an old slide. I'm now an associate professor. So the building that you see behind is where I am sitting right now. And this is IIIT-Delhi R&D building. So before coming here, I was in Singapore and I was a research scientist there. And I was there for 8-9 years and working primarily on genomics. So there I analyze different kinds of genomic data sets like epigenome, single cell expression, chromatin interaction, different varieties of genomic data. And there I realized the potential and the power of genomic data to make a long-lasting impact on society. So inspired by that, I came here. But before that, then I was in Finland, where I was 1 year in Malaysia. So I was asked by Dr. Saleh to actually talk about my career, just to inspire you people. So basically, I have to say that I am an electrical engineer from BTech. So I did electrical engineering, BTech, and And after that, I switched to computer science in MTech. And then I switched to computational biology. And today I can see that I am benefiting a lot from my knowledge in electrical engineering. Because if you see computational biology is much more complex than it looks like building data and simple running some ML tools. When you go deeper and start making your own tools for processing the data in the way that no other tools in the world does it, then you need such knowledge, and it helps. But don't be discouraged. It doesn't matter from which field you are. You should have. You can still contribute to computational biology. So, okay, so I'll start my talk. So, today I'll be talking about single cell epigenome profiles and what are the promises that single cell epigenome profiles can hold, and what are the challenges in analyzing single cell expression profile, a single cell epigenome profile, and how we have handled those challenges. So to start with, so in previous decade, a lot of data was being generated using bulk, and still it is being generated from bulk. There, the bulk sample had millions of cells. But now technology has advanced, and scientists have moved towards studying the cell at a single cell at a time. And there are different techniques to profile different profiles from the cells. I like single cell expression profile, single cell epigenome profiling can be done, single cell proteome profiling can be done, DNA mutation profiling can also be done, and lipidomic. And now the field is moving towards visualizing single cell level transcriptome and proteomic profile. So you can see here in the right side, you can see the expression of a gene, actually 3 genes, right, at single cell level, POL2R, POLR2A, PPIB, and UBC. So, and I am trying now very hard for single cell spatial epigenome. So basically spatial single cell epigenome profiles, which I will talk later, what are the benefits of that. So even though spatial single cell transcriptomics expression profile has not been so elaborate, like you cannot profile expression of like more than 10,000 or a few thousand genes, but it can be integrated with a normal single cell expression profile, RNA-seq, single cell RNA-seq. And it can also be integrated with single cell epigenome profile. I'm not going to that detail. The first, that is, my job is to make you aware of the single cell epigenome profile, the promises and the challenges. So single cell epigenome profiling can be done for different kinds of signals in the nucleus. So you know that chromatin scaffolds, and chromatin is consisting of nucleosomes and DNA, and these nucleosomes are made of histones, and histones can have modifications. So single cell ChIP-seq has been done in 2015, and that is to study single cell histone modification. Okay, then chromatin can interact, right? They can fold and interact. So single cell chromatin interaction profiling has also been done in 2013, but it captures only 2% of the interactions. So if you take 1 million cells and profile chromatin interaction, single cell chromatin interaction method will capture only 2% of what you get from a sample with 1 million cells. But in comparison to that, single cell chromatin accessibility has been very sensitive. And it really provides a view about where the chromatin is open, means the DNA is accessible. You can see here in the figure. So the nucleosome is loosely bound and the DNA is accessible. So you can actually, this shows that this part of the DNA can be bound by TFs and is accessible. And we know that DNA opens wherever there are regulatory sites, like enhancers and promoters. And then single cell DNA methylation is also there. It was done in 2013, first time. So today I will be mostly focusing my talk on single cell chromatin accessibility. So there is a protocol called single cell ataxia, which is easier to perform and can give in comparison to other methods that mentioned here, it is more sensitive in providing a view of the chromatin. So what are the promises of single cell epigenome over RNA-seq? Because single cell RNA-seq has really matured, and scientists are regularly using it for clinical samples. But you see what happens. I have been working with some clinical samples like circulating tumor cells. So these tumor cells are circulating in the blood, and when you use some experimental method to actually purify those tumor cells, these cells gets destroyed. The nucleus, or you can say the RNA, get really destroyed. So also in the all preserved samples, all cancer preserved cancer samples, a lot of RNA is already degraded. So in those cases, epigenome is very helpful. And epigenome is more preserved than RNA. And using epigenome, actually, you can predict RNA. The paper that I published in Nature Biotechnology, there I showed that just using histone modifications, 4 histone modifications, you can predict the mRNA level to actually 90% correlation using some tricks and techniques. So you can go through my paper in Nature Biotechnology. So, so it is possible using epigenome to literally predict the expression 1,000-year-old sample if it is preserved in ice or fixed using histone modification. For single cell, of course, there are still challenges. So, and another benefit is that you get better inference about the external stimuli, because we know that if you see 3 billion sequences in a human genome, only 2% is actually transcribing the coding genes, maybe even 1.5. The rest 99% of the genome is actually controlling the expression of genes, right? Timing and the place and the cell type where they should be expressed. Okay. So if a cell is stimulated from outside, it affects the whole genome. It doesn't affect only that the promoter of that 2% location. No. So these promoters are even just 0.1% of the genome. They're less than the promoters of the genes are so few, right? Like their number, their size is so less. But if you see, there are so many enhancers all over the genome that the external stimuli affects all of them. And if you don't measure those 99% genome, don't measure the signal in those 99% genome, you will not get a better inference about the external stimuli. So this studying non-regulatory, non-coding regulatory elements is very beneficial, and epigenome promises that. And since just 1% of the genome, that is the gene, right, it is very well preserved across all species. So actually, you don't get to know about the mood of the cell or the history of the cell without studying the rest 99% of the genome. Okay. And that's why epigenome gives you an idea about the mood or the potency, right? Mood, by mood, I'm saying the potency, the potential of a cell to differentiate to other lineages. You know it better when you analyze the rest 99%, right? Non-coding elements. Okay, so, and that's why researchers are now working aggressively on single cell epigenome profiling. Okay. So now, so just to give you an idea about how we pre-process the data of single cell epigenome profile. So you can see here, this is just for illustration purpose. So if you see here cell 1 or sample 1, and you see genomic bins arranged according to the number, so on a chromosome, and we know that by next-gen sequencing we get reads, right? So suppose we got reads on different locations, and we count those reads. So these looks like peaks, right? So so we do a peak calling, and we say that okay, which sites have peaks, right? Okay, and then on those peaks we count the total number of reads within certain window, like from the center of the peak, and we call it as a read count of that peak. So, and that peak can exist in multiple cells. So basically for each peak and each cell, we estimate the read count. So you get a read count matrix that is shown in the red. And then we start our business. Okay, so, and these read count matrices very huge in comparison to single cell RNA-seq. So in single cell, you know that single cell RNA-seq, we have mostly like 30 to 40,000 genes, okay? And they are fixed across multiple cell types, but it's not the same in single cell epigenome. Enhancers in one cell type might not be enhancer in another cell type, right? So, and because enhancers are more cell type specific, and in a cell, if you have like 30, 40,000 enhancers, and you have like suppose 10 cell types, so you get like 300, 400,000 enhancers, genuine locations that you cannot ignore. That's why these read count matrices, single cell epigenome profiles are huge. And they are also very sparse because the total number of reads that we get from next-gen sequencing for every cell is less, because the starting material is so less in single cell epigenome profile in comparison to expression. So there are some challenges. Okay, so and these challenges have been documented in a famous recent review by Lenneman et al. In Genome Biology, where they have talked about 11 great challenges in single cell data sciences. Out of that, 5 I have mentioned here. They're very relevant to single cell epigenome profile. That is, first thing is handling sparsity. There's a lot of places where you should get the signal, where you should have the read, you don't get it. So, okay, so you can't say that this 0 is genuine 0 or it should be 1 or there should be some read. And also, how to discover patterns in single cell epigenome profile read counts, and how to actually map single cell expression or epigenome profiles to reference atlas. So what do you mean by reference atlas? So different labs across the world have produced large atlases of single cell epigenome and single cell expression profiles. So now we have at least more than, I can say, 10 million cells, single cell RNA-seq profiles. For 10 million cells, we have single cell RNA-seq profile. And I can tell you more than around 1 million cells, we have single cell epigenome profile, especially single cell open-chromatin profile. So now if some lab produces new single cell epigenome profile, how it can match it to those existing data sets. So that is mapping single cell. That's a challenge in the single cell data sciences, because different labs have different batch effect. And then how to generalize the trajectory inference for single cell epigenome profile, and how to integrate data from different samples, experiments, and type of measurements. That's one of the challenges. Like, suppose you have a cell from human and you have another cell from mouse, okay? Same cell type. Okay? But when you take different cell types from human and mouse, okay, and try to cluster together, mouse will cluster one together, mouse cells will cluster one together, and human cells will cluster together because of the batch effect and the species effect, right? So there could be difference in species. There could be difference in the experimental batch effect. Right? The way, because there are different ways to profile a single cell epigenome. So these protocols may cause another batch effect. Okay? The sequencing depth by next-gen sequencing might be different across different labs, data sets. So that causes a huge challenge, and we have worked on it, and we have been very successful in that. So first, to start with, the first challenge that mentioned by Lanim and Tao, handling sparsity in single cell epigenome profile. So before we started this work, there were a few tools already for imputing read count matrices of single cell RNA-seq profiles. But as we told you that single cell RNA-seq profile read count matrices are still simpler than the single cell epigenome read count matrices, because they have fixed genes, fixed number of rows, okay, and less sparsity. And that's why the methods which we are working very comfortably with single cell RNA-seq could not work for single cell open chromatin profile. And we tried it. So you can see here magic. So if we take each dot represent a cell, right, and if we just use a famous tool called t-SNE for two-dimensional embedding, we see that different cell types mix. Similarly, when we use other methods meant for single cell RNA-seq imputation, they actually merge cells of different cell types to like with each other. So if you get a data where you have unrelated cell types, you will actually perform a wrong annotation. So this is not what we do, what we need, right? So we developed an algorithm for imputing single cell open chromatin profile with high accuracy. So we developed this method inspired by a machine learning method called random forest. So what happens? The basic idea is, how do we do imputation? So we do imputation by first saying that, okay, we have, like, suppose 1,000 cells. Out of these cells, these 5 cells look similar. These 5 cells look similar. This other 10 look similar. So what we do? Why not we just average their signal, right? So that's the first mistake. That is the first step where error can occur, because if you do wrong grouping of the cells, you do wrong imputation. And wrong grouping happens if the data is very noisy. And that's why what we do, at first we perform a very simple imputation on the first node, and then we perform a clustering. Okay. Then we divide the data into three portions, not the three portions, according to different clusters. Okay. Again, we use the raw data, and again we perform imputation. So we do imputation just so that we can get some cluster. Then in the end, we get smaller clusters and we perform imputation. So what happens, every cell gets into one cluster, and imputation is performed. Okay. Now what we do, every time we randomly, so while performing the clustering here, we don't use all 100% peaks, not all. We do it only for suppose 50%, randomly chosen 50%, and we try to find random number of clusters, like just 2, 3. So we use a randomization here. Similarly, because as I told you that we cannot believe on the clusters because data is so noisy. Even we do some imputation before, some basic available imputation method before clustering. So similarly, we do multiple times such clustering and imputation. Okay, so we build different multiple trees, and from every tree we get one or two imputed form of a cell, single cell open chromatin profile, right? So for original single cell open chromatin profile, we get multiple imputed versions. Then out of these, we choose those ones which are most correlated with the original one. Why? Because if the clustering of the cells has been wrong, the imputed matrices will be far away from the original. Okay, so we take the top 3 correlated imputed form and take their average, okay, and we build the final imputed matrix. And it helps. So we have phase 1 and phase 2, right? In phase 1, we perform imputation multiple times using multiple trees, and in phase 2, we assemble them. And this work has been published in NIR Genomics and Bioinformatics recently. And we see that now you can see here the final imputed matrix that we get. Cells are actually grouping according to their types, much better than the previous. And then one thing that we realized, the strength of our method, is that our method can handle imputing signal even from minor cell types. So often in single cell data from in vivo samples, like clinical samples, like take a biopsy from a cancer patient, right, and do single cell expression or epigenome profile. What will happen? You will see a lot of unbalanced data. So there will be like maybe very few cancer cell types and maybe multiple normal cell types, or there could be multiple types of cancer cells, and some of them might be just minor and rare cell types, but those might be important because if you give drug, other cells will die, and those minor cells then remain resistant, and then they grow again. Okay. So detecting rare and minor cells is one of the most important tasks in single cell, right? Because even FACS cannot catch it, FACS sorting. So because you don't know what are the markers of those cells. So we applied our method to in vivo data. So these cells were taken from the adult mouse liver. And what do we see? So first of all, we see that, okay, hepatocytes are 91%, very unbalanced data. Endothelial cells 3.2%, endothelial type 2 cells 2%, and T cells are just 0.2%. We applied other methods. And if you see down, we also applied some other methods meant for single cell epigenome visualization. Not only that, even Scale does imputation also. Scale and SC Open, they were proposed after us. So we were the first one to actually propose to apply single cell epigenome profile imputation. And after us came Scale and SC Open, but even they could not recover the signal of minor cell types so that they could be grouped or clustered well. But if you see FITS is our method, this one. So FITS, we can actually recover the signal of minor cell to such an extent that they can subcluster together, like according to their own type. So this was very encouraging, and it paves the path for better analysis of clinical samples using single cell epigenome profile. Okay, so coming to other grand challenges in single cell data sciences. So as I told you that there's one grand challenge of mapping the existing, mapping your data to the existing reference single cell atlas. So just to inform you that the work that I showed previously was actually done with the help of two students here, Nitesh Pandey and Rajesh Sharma. And the third challenge mentioned here is being handled by Shreya Mishra, who is a PhD student in my lab. So now, what are the benefits of mapping your single cell profiles to reference atlas? So first thing, that you take benefit from published data. Second, you reduce redundancy in producing new data set. Suppose suppose you want to study a developmental time point, right, in months, and you performed very simple experiment. You got 100 cells, but when you match it to the existing atlas, and you say that, oh, those cells are already matching to these cells, that there is no need to produce this data set. Maybe I can save that money for a special case of developmental time point. Maybe if I put a knockdown of a gene and then perform the experiment, and the other existing data, I can use it as a normal. Right? So you can save the resources, reduce redundancy. Okay, but the problem is how to actually assemble all this reference atlas data at one point, at one place, right? And how to actually search them, how to store this huge single cell profile, because you have 10 million cells. I'm talking about expression. 10 million cells and 30,000 genes. And single cell epigenome profile, every lab read count matrix of single cell epigenome profile is different. They have different peak lists. Each one has like 100,000 peaks and maybe 100 to 700,000 cells. So there is more heterogeneity there. So how to store them, and how to use that for studying cancer cells or annotating new cells. How can we use that to find that whether the cell that we get from a biopsy of a patient, whether that cell is going to die from a drug, or whether they will develop drug resistance, whether they will differentiate on application of certain drugs, right? That is potential. How to determine that using the existing data set. That's why a search engine is needed for storing those huge single cell profiles and accurately matching the cell states. Okay, and imagine this, how it will help in cancer biology. So you take a patient, or maybe just old sample, 10-year-old sample from a cancer biobank, and then you perform single cell epigenome profiling, and then you want to study the cellular state. Why this patient didn't respond to this drug, or why this patient actually responded to this drug. I want to study like 10-year-old story, because that will build knowledge, right? Single cell epigenome profile can help you, because samples are 10-year-old. Single cell RNA-seq cannot be done because RNA is already spoiled. But using single cell epigenome profiling, you can do that. And by matching them to reference atlas, you can actually further use existing knowledge. So, how do we get this idea? So actually in 2018, we published the world's first search engine for single cell expression profile. So it was published in Nucleic Acid Research. And it was kind of primitive, but it was world's first. So then after publishing that single cell expression search engine, we said, okay, now maybe can we do it for single cell epigenome profile also? But there are challenges, because if you see single cell RNA-seq profile, you have same gene list across multiple labs, data from multiple labs. But using single cell ATAC-seq, if you have data from two groups, they have different peak lists, right? And so that's a challenge. And read count matrices are also very large and sparse. So these are read count matrices, they are very large in comparison to single cell expression profiles. And of course, there is batch effect and read count depth problem, like every cell may have different read count. How to handle that? So we also made probably the first search engine for single cell epigenome profile. We call it SAP Search. So it's already there on the GitHub. Not GitHub. It's already there in bioRxiv. It's under review now. So, but you can start using it right now. So how do we tackle the challenges in SAP Search? So suppose we have read count matrices from two different groups. So where you have peaks in the rows and column represent cells. We first calculate gene enrichment score. So how do we use, so you say that, okay, those peaks are not lying on the gene, how will we calculate the gene enrichment score? So what do? Actually, we take the most proximal gene, especially for single cell ATAC-seq. So what do we do? First, we use very simple rule of epigenome, that enhancers, so genes near enhancers will be influenced by the activity of that enhancer. Okay? So we take proximal genes. First, we highlight enhancers from open-chromatin profile by normalizing them with global accessibility score. Then we get their cell type specific activity score, and we know that enhancers are cell type specific. So once we normalize the read count matrix, we take peaks with high normalized read counts. We take their proximal genes, because genes near to the active enhancers will have higher expression. Okay, and then we convert that read count matrix to gene enrichment score by using statistics like how many active enhancers are proximal near this gene, right? Suppose a gene has 4 active enhancers around it, it is more highly likely to be active than a gene which has just 1 enhancer. Okay? So that can be done, calculated using Fisher's exact test. So once we do that, we solve the first problem. Then the question is, we have converted single cell epigenome profile from different labs to gene enrichment scores where you could have same gene list. But now you will say that, oh, now we just correlate, right? We just correlate and then it will be done. No, it will not work. So we have tried all that. You say that, okay, we will do all this machine learning methods will apply, autoencoder and all that, that will not work. Simple matching steps will not work in this case, because data sets from different labs have different biases. Again, we use the same method, same approach, which is being used in the field of epigenome, that the genes near active enhancers will have higher expression. So what do we do? Suppose a query cell came. Okay. So we have somebody submitted a single cell epigenome profile of a cell. So we found what we take. We take this top 1,000 enriched genes. So we convert it to a gene enrichment score. Then we take a top 1,000 enriched genes, and then we look for. Now suppose we have to match that query cell single cell ataxic profile to existing expression profile. So we take this median expression of this top 1,000 enriched genes and in every cell in our reference. So suppose our reference has like 10 million cells. So we try to calculate the median expression level for those 1,000 enriched genes in all 10 million cells. And the cells which have highest median expression level of this top enriched genes is most highly likely to be having the same cellular state as the query cell. Similarly, we do it this way, for median. So here we just use some, since we have already converted the single cell, so in the existing single cell open-chromatin profile into gene enrichment score. So rather than calculating correlation, we just use the same way like we did it for expression. We calculate the median enrichment score of top 1,000 enriched genes. This way, we are able to avoid the batch effect, and it works. And we have tried autoencoder, we have tried correlation. Normal machine learning method doesn't work. So at some point, that's why I told you that knowledge of biology helps you. So if you embed it properly with machine learning methods, it works. It does magic. And then you can see here that we get very high accuracy. So we tried it with different with open chromatin profile query from different cell types, and we get like almost 100% accuracy in finding the right match. Okay. So now, and we could also annotate certain unknown cells in the published data from mouse thymus. So there was published data from mouse thymus, but authors could not annotate some of the cells. So we actually annotated some of the cells by determining their type. And we know that thymus actually is involved in white blood cell production. So you see white blood cell is coming on the top, it produces T cells. So it's quite accurate. And so, but determining cell type is not enough, right? How do we use it for studying potency? So we did that. So basically K562, a cancer cell, is known to be multipotent. So mainly it is erythroid-like. So what we did, we took the matching scores of the query. So you can see here the heat map. So basically every row shows a cell, query cell, K562 query cell, and every column shows the query hit, or you can say that the matching cells. So we calculated their match scores, and we take it as a matrix, and we perform hierarchical clustering. And what we get? We got two clusters. So cluster 1 cells, we found that they are highly multipotent because the top enriched genes, they actually oppose erythroid-like phenotype when hematopoietic stem cells differentiate. So They push the cells towards megakaryotic lineages. So we found cluster 1 to be enriched with phenotypes of megakaryotic lineage. Just so you can see how epigenome, if you do it at expression level, you may not get this. But as I told you, that epigenome keeps the hidden signal. So epigenome was able to highlight the heterogeneity within these cancer cells. And it also explained why these cells are multipotent. So in these cells, some cells were actually poised for other lineages because they de-differentiated to progenitor states. And we found that using our SAP Search engine. Now, coming to the next challenge is that is very challenging even for computer science guys. Even the previous problems were challenging, but this is most challenging because the challenge is that we have single cell data sets from different species, different experiments, different labs. And now you have to integrate them such that you should be able to group same cell type together. That's a challenge. And SAP Search does that very, very nicely. So you can see here, there are 4 methods shown here, published in very reputed journals like Nature Communication Methods. So they were meant for single cell expression profiles. We call them as a embedding. And so we took human hematopoietic stem cells, mouse hematopoietic stem cells, mouse neurons, human B cells, and human GM128. That is also a B cell. Right? Human GM128 from different lab. Okay, so there are 2 different. So there is single cell open chromatin profile of GM128 cells from 2 different labs. Okay? And then we have human myoblast cell. Okay. Now, taking their single cell epigenome profile, then we give them to those data sets or read count matrices to other tools. They just mix them all. Okay. So you can see here, they are either they are not able to group same cell type together, or they mix them wrongly. Like Canorama mixed completely wrongly. SCVA5 completely failed. They grouped them separate. But SAP Search, you can see here, human hematopoietic stem cells and mouse hematopoietic stem cells are coming together, right? And then human B cells and human GM cells, they are coming together, and the human GM2 other batch also coming together. So basically, irrespective of species and batch, we are able to cluster them together, and that's the power of that's we call a powerful search engine, right? No matter what is the batch effect or species, we find the correct cell type for you. And we are trying to now going to the next level of this challenge is can we find the disease state, correct disease state of the cell? And the challenge there is that we don't have enough. So as I told you, the single cell field has to still progress. And so there are huge consortiums which are trying to actually profile expression and epigenome profiles of cells in diseased state or from different tribes. So in future, you may see billions of cells profiles. So you don't be surprised if you see single cell expression profile from billions of cells. So we are making the second version of our first search engine. where we will have 10 million cells and from different labs. Okay, so now the last challenge, that is generalizing trajectory inference. So what is trajectory inference? This is being handled by Smriti Chawla. So she published our solution in nucleic acid research recently. So what is trajectory inference? Trajectory inference is that suppose you have growing embryo, right? And where every cell is like poised to go towards different lineage, and you take suppose 10 mouse embryos, just very early stage, and from there you harvest 100, 200 cells, you do single cell expression profiling or ataxic profiling. Now you want to see which cell came first and which cell came later. So that is trajectory, right? So which cell came first? So basically finding the temporal ordering of the cell. So that is, we also call it temporal ordering of the cells. So because temporal ordering of the cells can actually give you much better insight about the grouping and many things. So I cannot talk about this right now. So, but before us, there were very few groups working on trajectory inference using single cell epigenome profile. So there were many groups, there were like 50 plus methods for trajectory inference or temporal ordering of single cell expression profile, but not single cell epigenome profile. So because Saleh et al. Has said that for transcriptome data, the features are well annotated. Okay, the features are genes, right, and correspond to expression level of genes. But for epigenome profiles like methylation profile or chromatin accessibility profile, what happens? Features are not well defined. As I told you, that every cell has its own peak list, own enhancers. Every cell type its own enhancers. So how to handle that? So again, we solve this problem by transforming single cell epigenome profile to pathway space. So you might have heard about gene set enrichment analysis. So we did kind of a single cell gene set enrichment analysis for using single cell epigenome profile, where we convert the epigenome profile of a cell to pathway scores. Okay. Again, we highlight the enhancers, and then using active enhancers, we use hypergeometric test, and then we calculate the p-value of enrichment of different gene sets, that is pathway. Okay, and the benefit of that is that our approach, we use a global accessibility approach. So using our approach, we get highly consistent results. So you take, for the same cell, you take neighboring cells, you group them with other cell types, right? And you use other methods. You may not get the same pathway scores, but in our case, you get a very consistent pathway score, no matter what are the other neighboring cells. So we handle each cell independently, and that is strength, because in single cell, if you can't handle each cell independently, there is no benefit of single cell. Right? If you mix it as a signal by other cells, so it will be influenced by neighbors, right? So, but there are other challenges also using single cell open-chromatin profile. Like there is a challenge for predicting chromatin interaction using single cell open-chromatin profile of the single cell epigenome profile. So how do we do that? So, so the rule is simple, that if there are two sites interacting with each other, right, there are high chance that they will be coactive. There is high chance that they will have open chromatin accessibility of same level. Using that, so there was a previous method called Cicero, which uses co-accessibility to predict chromatin interaction. But we found that Cicero is not able to predict distal interactions. So if there are two genomic locations beyond 500 kb, so we developed a method, locations like around 1 MB or more than 1 MB. So I hope you can hear me. Or
Yeah,
yes,
Dr. Vibhor, we can.
So our method, you can, so it is still under review. So we call it SC Chia. It is more sensitive for distal interactions. Now the question is, and okay, so I'll skip this slide. Okay, let me first conclude, make the conclusions of our work, and then I'll go to some more interesting higher level question. So first, we are able to handle sparsity. We are pioneer in doing that for imputing single cell ataxic profile. And then we are pioneer in mapping open chromatin profile to reference atlas. We are pioneer in transforming single cell epigenome profile to pathway space and using it for clustering and temporal ordering. And we were not pioneer in predicting chromatin interaction using single cell ataxic profile, but we have improved it. We have improved the previous methods. Now, okay, so these are all technical things. Now, one of the higher level questions that can be answered using single cell epigenome profile. I mean, why this craziness? I mean, why do we waste so much money in next-gen sequencing for single cell open chromatin profile or single cell epigenome profile, right? So as I told you in the beginning, that when you have circulating tumor cells in the blood, so liquid biopsy, as you know, is getting very popular. So you have CTC, circulating tumor cell, is one of the type of liquid biopsy where cells are purified and their expression profile is taken. So you will find a lot of papers on expression profile of CTC. But don't be surprised if you see the strength of that is that CTCs, mostly when you try to purify, the RNA is destroyed. And you have to have very expert hand to purify the CTC as well as keep this RNA intact. But for single cell epigenome profile, you don't have to worry about it. DNA epigenome is more preserved, right? So first of all, studying old samples, older cancer samples, CTCs is perfect. Then our lab is currently working on cancer therapeutics using single cell epigenome profile. Now you will see how we can be done. So as I told you that we can predict chromatin interaction, right? Using single cell open chromatin profile. So if we get biopsy sample of a patient, right? We take out cells from there, we do single cell epigenome profiling, okay? We can predict chromatin interaction. So from the same epigenome profile, we can find active enhancers in every cell, okay? At the same time, we can find chromatin interaction in different groups of the cells. Then we know that which enhancer is targeting which genes in different types of cancer cells. Okay, and we can target those enhancers rather than just targeting the genes. We can target those enhancers because enhancers are more cell type specific. So this will have least side effect. So we are working on on developing methods to cure cancer with least side effect using single cell epigenome profile. And also,
sorry, Dr. Vibhor, I'm sorry to interrupt you. We are exceeding the time, so if it is possible. We are exceeding the time, so if it is possible.
Yeah, so last slide.
Yeah, sure. Thanks.
Oh. So, and what other things to expect in near future? So first thing that we are also working on, predicting the future state or the potency of the cell, like drug resistance, right? So, and also developing new differentiation protocols. So as I told you that we can transform the open chromatin profile, a single cell open chromatin profile to pathway space. Okay? Then we can study which pathway is pushing the cell to which lineage, right? So we are working on that. So in the hope that we could develop new differentiation protocols where we can actually just perturb the external pathways and guide the cell to desired lineage, right? And that's what we profile, because epigenome gives a better picture of the potency of the cell, right? So we can perturb pathway and we can say, oh, what happened to the potency? Which lineage is poised towards, right? So so we are working on that. So developing new differentiation protocol, finding drug resistance or future potency of the cancer cells. So now I would like to acknowledge my PhD students. So Nitesh Pandey, Rajesh, and Shreya, and Smriti, and Omkar, and other collaborators from GIS like Tam Wailong and Sally Kong, and one of the collaborators, Devaka Sen Gupta. So I'm open for question now. My talk is finished.
Yeah, thank you, Dr. Vibhor. So there are a few queries which are needed to talk. One is epigenetics has also explained, epigenetics can explain the concept and pathway of neuroplasticity.
Neuroplasticity.
Yeah.
See, neuroplasticity has different dimensions. Neuroplasticity, I had this question in my mind a few years ago. I still think neuroplasticity has different dimensions. Just like you see, cancer plasticity also has different dimensions. So yes, some portions of neuroplasticity epigenome can answer. That's just like, you see, there are three things where epigenome is involved, just like in your vaccination. When you get vaccinated, right? So, that's a memory in your immune system.
Yes.
Okay. Then second, the memory in your neurons, like to some level, it controls the plasticity. And the third is plasticity of cancer cells. To some extent, yes, but neuroplasticity is a broader topic.
Okay. So there is one question. How this epigenetic studies can help in post-COVID era of genomics, or is there any reference? Is there any reference where COVID is associated or linked with epigenetic studies? Is there any study already ongoing in any lab or somewhere?
Yeah, that's a really good question. Okay, so for that, let me share a screen. We actually published. I have not talked about the paper. So let me see we published our papers recently in briefings in bioinformatics. So where we actually so this is on bioarchive, but this is also already published in briefings in bioinformatics. So let me go to the last figure that will give you an idea about it. Okay, so in this manuscript, what we do is that we are using single cell expression profile. I will link to his epigenome profile later. So we use single cell expression profile to infer gene networks in old and young lung cells, right? And then using the inferred gene network, we found what are the genes which have more degree, or you can say gene network based importance, like influential in affecting multiple genes, like in the network. If in old and young mice lung cells. And then we did perform gene ontology. We found that in older mice lung cells, and those pathways which are enriched for the most influential genes in the network were actually involved in T cell activation, inflammation by cytokine, RAS signaling pathways, and others. Okay, and JAK-STAT signaling pathway.
Okay.
And we could not get some of the pathways just by differential gene expression among old and young. So by network inference, we could get a better idea. Then we compared, then we took lungs data, or we say lung expression data from patients affected with COVID. Okay. We performed differential expression. We found which genes are upregulated in lungs with COVID. Okay. We did the gene ontology of those, and we found the same pathways coming up: JAK-STAT signaling pathway, RAS signaling pathway, inflammation. We found same pathways coming up, which were coming for older lung pneumocyte 2. So those lung cells were actually pneumocyte type 2, which are the prime target of COVID. And then, so why I'm showing this? What I'm showing is that gene network inference, right, can be done very well with single cell, or you can say epigenome profiles. So, because you know the direct target, if you know, so epigenome profile can help you in gene network inference. And gene network inference, once you infer the gene network, you can actually make multiple conclusions, just like in this paper.
Yeah.
So I hope I have answered your question.
Yes, certainly. So what will be the fate of epigenomics in post-COVID era? Means once the COVID is over, there will be, means we may have plethora of studies around epigenomics. And
cancer is something that will always exist, right? Okay, and yeah. Regenerative therapy is something that will always be put there, right? If somebody has spoiled his kidney or lung cell, right, and we want to regenerate certain organs or certain tissues in his body spoiled by infection, it is always there. That's evergreen field, right? And there, as I showed you this figure, that we can use a single cell epigenome profile to find new differentiation protocols to achieve that lineage. Suppose we want to achieve a particular cell type from embryonic stem cells. So suppose we take a person's skin cells and we want to transform it to some other cell type. In order to do a regenerative therapy, like to replace or put some cells in the organ which was spoiled, right? There, you need to develop new differentiation protocols, because differentiation there are differentiation protocols for very few types of cells, right? So that regenerative therapy needs it. Okay.
Sure. So one last question I would like to ask. Smoking influences the DNA methylation pattern.
Mm.
And can this be used, means epigenomic studies can be used for drug abuse in sports persons, sportsmen and women, something?
Yes, it can be. Yeah, definitely. Yeah, that's a good question. That if a person is using drugs, in fact, so drugs is what? I mean, it's just a chemical, right? It's just like other drugs, right? So,
Yes.
so that is being regularly done. If you read papers, there are scientists who actually give drug to mouse. So there could be not there's additive drugs, but there could be just therapeutic drugs, right? And they study the effect of drugs. And soon we are going to send a paper in Cell where we have applied the most famous antidepressant Prozac to rat. And so basically what we did, we had two groups of rat. When one was given Prozac, other was not given Prozac. And we took out their brain and studied their 27, and then we micro dissected 27 regions from their brain, and we profiled epigenome as well as single cell expression as well as bulk expression, and we made multiple conclusions. So we are sending the paper, might be soon out on bioRxiv. So of course, that's
Sure.
a hot topic right now.
So that was the last question. Thank you all for joining us for very interesting and informative webinar. I really thank Dr. Vibhor for sparing time from his busy schedule for such an interactive talk with prime focus on analytical challenges associated with single cell epigenomics. I request all participants to fill their feedback form for today's webinar, today's talk. And those who have attended webinar through Facebook Live and YouTube Live, they have to fill their feedback form, which will be there in the description box. And those who have attended through Zoom, they also might have received their feedback form for this. And do carefully put your name in the feedback form, because it will be used for generation of certificates. I would also like to inform all participants that in the series of webinar, we will have Dr. Koel from IISER Kolkata in the next talk which will be on 5th of August, where she will be discussing about group discussion using neural data fusion. For more information about this webinar, please keep visiting Mano at social media platform at Twitter, Facebook, and Insta, and requesting all to keep visiting the social media platform for regular updates. I also thank outreach team of Mano to organize this webinar and whole technical team for their support. Thank you all, and good evening.